Thank you very much. So I was asked to talk about my 5 selfish reasons to work reproducibly, and I looked at my talk again and I discovered 2 things. A, my 5 reasons had become 7, and the whole talk took more than an hour, and I only have 20 minutes. So I've cut it down, and I will still talk about a very selfish approach to reproducibility, but I will focus a bit more on my own thinking, how I got to take this approach and why I'm taking it in contrast to other approaches to motivate people to be reproducible. So I think it's fair to say the reason many of us are here is because we don't know how to get to the next slide. Because we're all very frustrated. So many of us are frustrated. What are we frustrated about? Well, there's all these flashy papers, not more rigorous work, nothing is reproducible, papers cost a ton. I mean, my last Nature paper, £10,000. Academic salaries are always as crap as they have been. So people concluded that everything is broken. The review system, broken. Publication system, broken. Job market, broken. Science, broken. Everything's broken. This is where this open scholarship and open research movement comes in. It is in big parts a reform movement that says we should be able to do better. And part of how we should be able to do better is sharing more, having better access to papers, sharing the source code of our analyses, sharing the data, sharing educational tools, sharing everything to develop science in a way that is more equal, more diverse, and more inclusive. Now, one of the big players in this open research field in the UK is the UK Reproducibility Network. And for my sins, I am the local network lead of UKRN here in Cambridge. UKRN is UK-wide. Started in 2018, so it's pretty young. You see on the map, I mean, it's somewhere here, it says 15 members. No, it's many, many more. You see all these dots. There are 2 types of dots. The small ones are places like Cambridge, where they have a local network lead who's more or less active. And the bigger, darker dots, these are universities that have invested much more, and they have actually given some money to support senior academics to lead these endeavors. Our local version of UKRN is currently called RRChem, and many of the speakers here of this session today participate in there. If you are interested in reproducible research in Cambridge and want to be involved in this, shoot me an email. My email is fm327@chem.ac.uk, and I can get you involved and would hope some of you, especially younger people, currently we're a bit professor heavy, which is not very good. There should be many more ECRs. People becoming involved and then helping us to set up a grassroots movement here in Cambridge based around reproducibility. Okay, with that as a start, for me, what is the reproducibility issue? Well, you see it here in this cartoon, which I'm sure you have seen before. You see these 2 gentlemen in front of a blackboard. I guess they are mathematicians or theoretical physicists. They have some very advanced maths on the top left, some very advanced maths in the bottom right. And in the middle, one of them has written, then a miracle occurs. And one says to the other, I think you should be more explicit here in step 2. And to me, this is how it feels when I read a big paper. There's some complex project on one end. There's a fancy paper in Nature or Science at the other end. And how did they get there? How did they take all these data, all these results, and cut them down into 8 pages and 4 figures? Well, there's a miracle occurred, and the miracle has something to do with data, with code, and with methods. And I don't want miracles in science. I want to understand how people get from their complex data to their paper, which is why I want to see their code and their data and understand their methods. And for me, this is the kind of transparency and reproducibility I want. I have pretty low standards. I mean, I don't want one-click reproducibility. If you share your code with me, I'm cool with that. The wake-up call for me was a story in my field. I'm a cancer researcher, and that is a story that features these 2 people here. They are biostatisticians. Biostatisticians are not the rock stars of science. And even so, these 2 made it onto the front page of the New York Times 10 years ago with a story titled How Bright Promise in Cancer Testing Fell Apart. So their names are Keith Beggerly and Kevin Coombs. I think they still are, at least back then they were, biostatistician at MD Anderson. The story I'm telling you is not at MD Anderson, it's at Duke University. Duke is in North Carolina. And it's a story about cancer research, which just proved completely irreproducible. And in my community, that was the wake-up call that made many of us aware of the problems. So the story starts, and these are slides I took from Keith, with very visible papers in like the top journals, the New England Journal and Nature Medicine, where people from Duke, a group led by a researcher called Joseph Nevins, the story here is the reason he went into early retirement, reported that they had done something really cool. They had taken cell lines. So cell lines are cells that live on plastic, and they treated them with drugs. They measured how the genes, the expression of the genes in the cell lines changed. And based on these gene expression changes, they could predict in patients whether the drug would work or not. So they went all the way from cell lines, artificial systems living on plastic, to making predictions about real human beings who need the best drugs to cure their cancer. This was amazing. So obviously the MD Anderson people said, we want to do that too. And because a big chunk of it was statistics, they went to the biostatisticians and said, please, can you figure out what the Duke people did, because we want to have what they're having. So the MD Anderson biostatisticians went to the papers, downloaded the data, tried to run the code, and completely and utterly failed. They couldn't reproduce anything. So I give you a couple of problems they ran into. One is called, well, an off by one error. So forget the R code you see here, but you see there are two lists in yellow. One is called theirs and the other one is called ours. So theirs is a list of gene names. Think of these funny things like 1881AT. Think of those as names of genes, which is pretty much what they are. They are from a measurement technology called a microarray. It doesn't really matter. These are lists of gene names. And theirs is the list that Duke had provided. And ours is the list that the MD Anderson people had tried to reproduce. There was absolutely no overlap between the two lists, but there was a pattern. If you look at the numbers here for a second, you will see one of the lists from Duke says, one of the names is 1881, and what MD Anderson found is 1882. Duke found a gene name called 31321, and MD Anderson found one called 31322. So they're not the same, but they're just shifted in a list. There's just a difference of one. How did that happen? Well, Duke used the devil's tool. They used Microsoft Excel. And so at some point, they had to copy and paste their data and gene names into a statistics tool. And the list of gene names had a header, which said gene names. And so they copied that with the rest of the gene names, and so everything shifted by one. They didn't notice it because by good luck, their gene names were all important DNA damage response genes, and it made a lot of sense to see them. Whereas the list from MD Anderson is all random genes, but they are the real ones. This is something that is not fraud. This is something that has happened to everybody who has ever analyzed data. It's just in Duke, nobody spotted it because nobody saw the analyses except for the first author. But there were other problems. So, okay, that's a busy one, but I told you the Duke story is about cell lines and how they respond to drugs. What you see in the plot here are what's called the NCI60 cell lines. It's kind of the way people overrepresent their results because the NCI60 cell lines only have 59 cell lines and no 60. But what you see on the x-axis, on the y-axis here are these 59 cell lines. And you see on the x-axis all kinds of different drugs that the cell lines have been treated with. There are blocks for each drug because what the biostatisticians have done is they color-coded whether the Duke people had reported the cell line to be sensitive or resistant to a drug in different papers. Now, it's a feature of this combination of cell line and drugs, whether it's sensitive, it shouldn't change from one paper to the other. So this plot should be very, very boring with everything being red or everything being blue per drug. But you see this kind of blue-red checkerboard pattern because the Duke people had just, in a way, randomly sometimes called it cell line sensitive and sometimes called it responsive to a drug, which is a very weird thing to happen unless you really want to have very small p-values, since they have to, anyhow. This was very weird. So it turns out the MD Anderson people wrote up what they found in the paper, which is very famous in my field. It came out in the Annals of Applied Statistics, which is one of those very technical journals that nobody reads, but this paper has been read very widely. It coined a term called forensic bioinformatics. Forensic bioinformatics means that you do bioinformatics, so you analyze biological data, you see something went wrong, and you try to figure out what must the other people have done wrong to get this incorrect result. They list whole kinds, lots of things in there, and they corresponded with the Duke people and said, look, we can't reproduce what you're doing, can you help us? And the Duke people, they didn't really want to, but at some point they had to help. But whatever they did, there were still problems with the results. And part of it was because turns out the first author was actually a fraudster. And so the first author of all these papers is Dr. Anil Potty, who also lost his job and left science. So it sounds a bit like this is a story about fraud. The first time I heard about this was here in Cambridge in my own institute in a workshop I organized in 2010. And so this is Keith Baggerly here in my institute lecture theater, talking, like giving a very funny talk about the kind of problems they had found in this analysis. And in my institute, everybody was laughing. The whole room was full of people laughing about the stupid Duke people and all the mistakes they had done in their analysis. Me and my friend sat in the back row, we were not laughing. The reason we weren't is because we were working on a very big project at that time, which later was published in 2012, on 2,000 breast tumors and a big data integration analysis where we looked at the genome and the transcriptome. It was a huge project. And we listened to the stories from Duke and we thought, okay, we don't think we have a fraudster here, but many of these problems can happen to everybody. Like the off by one, this could just happen to all of us here. Even as an honest mistake, even if just everybody tries their best to work well. So how do we avoid running in all these problems? The problem with Duke for me really is lack of transparency. The reason the fraudster could pull off these tricks is because nobody but him ever looked at the data and the analyses. And these were small data sets. I mean, the NCI 60, I mean, these are really small. Nowadays, you can analyze them on your cell phone. Whereas what we had is still a huge data set. And so in the end, what we decided is a strategy which really depended on code and analysis sharing, having documented markdowns of every step of the analysis, and always having groups of people look at the analysis to make sure there's never just a single person who signs off on stuff. And for me, this was my wake-up call. So a few years later, I then wrote a paper called 5 Selfish Reasons to Work Reproducibly, because I had profited so much from working reproducibly that I thought I should share this with the world. And it's really about what's in it for me approach to reproducibility, because whether you like it or not, it's a big investment. You have to learn tools, you have to learn programming. Working reproducibly is hard, but I believe it's worth it. So my 5 selfish reasons were, well, first of all, you avoid disasters. If you work reproducibly, you do not have to go into early retirement like Joe Nevins because one of your postdocs messed up. I think it makes it easier to write papers because every time you look at the draft and something changed in the data, all the p-values might have changed too. So you really want annotated, markdowns, easily available, reproducible documents that tell you the newest numbers. In the paper, I tell you an example where I had very good experience with a reviewer engaging very deeply with our papers. Only because we had shared them in a way they could engage with in a nice markdown, had shared the data, and we had a very constructive discussion about how to analyze the data. So people complain about the review process being broken, but because we offered the reviewer something, reproducible code, we had really great experiences. Some of it is just continuity. You profit because you still remember what you did 4 years ago. And it's very good for your reputation if you're known to be a trustworthy and reproducible scientist, and all of that is good for your career too. Now, this particular selfish approach, the reason what I liked about it is different from other approaches people use to convince you to work more reproducibly. One of them is what I call the preacher approach. So people stand there and say, well, why should you work reproducibly? Because it is the right thing to do. Because it's the foundation of science. It's honorable. And it's making the world a better place. And I don't know. it doesn't, I mean, it's not like this is wrong, you know, but the whole making the world a better place thing, I mean, this sounds a bit weird to me. If you saw me here, I mean, that's not how I look. I mean, I don't even wear sandals. I'm not in this game to make the world a better place. I'm in this game to win it. And so working reproducibly is for me something that, well, I want to get something out of it. So my question was, ask not what you can do for reproducibility, ask what reproducibility can do for you. And this is a very different approach why you want to engage with it. In the selfish view, you engage with it because you get something back. Now, the second view of reproducibility that I really do not like and that I want to have a counterpoint to is what I call the replication police. And these kinds of replication studies that Alexander showed, they left a bad taste in many people's mouths because it sounded like there's this outside force coming in, checking whether I'm reproducible, yes or no. It's like Judge Dredd coming in, telling me he's judge, jury, and executioner in one person and judging my research. I mean, of course, people were defensive about this. And this is, I don't think you motivate people like that. If you just say, well, if you don't do it, Judge Dredd comes. I mean, I find the selfish approach where I help myself, I find this much more convincing. In particular, because I do not believe that reproducibility is just a binary black-white thing where you're either reproduced or you're not. So the whole, it's not a question of life and death. It's a very grayscale thing where some parts might reproduce, some might not. And if they don't, the interesting question is, what is the problem? Why did it not reproduce? I mean, is it because people didn't know what they were doing? Is it because the reagents are somehow fragile? Who knows? I mean, it's really interesting to figure out why reproducibility failed. So following up on this, we just published a paper very recently about different reproducibility standards for machine learning in the life sciences. And we tried to capitalize on the idea that it's not black or white. So the standards we propose are bronze, silver, and gold. And the lowest level bronze, that's achievable for everybody. This is just, you give me your data, you give me your model, and you give me your code. You don't even have to write it up in a way that's easy to read. You just give us all of this stuff. Now, if you're nice and you write it up in a way that's easy to read, you're already at silver. And if you finally give it to me in a way like a Docker container or whatnot, that I just push one button and the whole thing runs, that is gold. In my own work, I find a bronze is a given, and I try to get my students to get to silver. We have never even attempted gold. Some of my colleagues are a bit more ambitious than me. But you see, there's a whole range. And you just have to find yourself on this range. All right. Now, finally, because I think I'm running up against the end of my slot here, one of the reasons, the reason I think about reproducibility and data sharing and all this, there is a deeper, more fundamental reason, because I want science to be trustworthy. And so why do we trust science? And I followed this book. There was this book here I read, because I wanted to know why to trust science. And there's by Naomi Orestes, there's a book on why to trust science. So I read the book. Her answer to that is that trust in science builds on a diverse community of experts critically vetting all the evidence out there. And I was thinking about this, being an expert myself. How can I actually critically discuss all the evidence out there? Well, it only works if people share the evidence. So I need to have all the evidence in my field, means data and code and analysis scripts. And I need to have all this evidence openly accessible to everyone. And this is why open sharing of code and data is so important for me, fundamentally important to any kind of trust in science that I can have. So with that, thank you very much for listening to me. And I hope we're having a good discussion about that. Thanks.
Hello, everyone. Hello from me too. I'm Dr. Maria Tapali. I'm an affiliated lecturer in psychology and education based at the Faculty of Education here at Cambridge. I'm also the Deputy Director of Instruct Research Group, which is a cognitive psychology applied education research group led by Professor Michelle Ellefson. So the past 4 years, once this open science movement started, the lab started getting interested in that, and we started doing trainings and seminars and trying to learn more about open science and what could be some of the techniques and strategies that we could implement in our own workflow, in our own research projects, so that we make our work more reproducible and more transparent. So I'm going to talk about some more practical steps now. We have covered the reasons why it's important to engage with reproducibility and engage with the open science movement. But now we will switch gears a little bit to think about how we can do that. I should note that this is just a mere introduction to that, because in 20 minutes I cannot cover these topics in depth. So we're going to start with only a few slides on introduction to reproducibility, just a few slides to get the talk started. And then we're going to move to talk about practical tips for reproducible quantitative research. And finally, we're going to talk about transparency and practical issues in qualitative research. I should make a note here that I'm a mixed method researcher, so I'm interested in both types of research, their distinctions, and how I can make my work more reproducibility with both quantitative and qualitative data. So what is reproducibility? I think you have a pretty good idea of what it is by now. It means that research data and code are made available so that others are able to reach the same results as are claimed in the scientific outputs. So improving reproducibility leads to increased rigor and quality of scientific outputs, and thus to greater trust in science. There has been a growing need and willingness to expose research workflows from initiation of a project and data collection right through to the interpretation and reporting of the results. So these developments have come with their own sets of challenges, of course, including designing integrated research workflows that can be adopted by collaborators while maintaining high standards and integrity. So the concept of reproducibility is directly applied to the scientific method, the cornerstone of science, and particularly to the following 5 steps: formulating a hypothesis, designing the study, running the study and collecting the data, analyzing the data, and reporting the study. So each of these steps should be clearly reported by providing clear and open documentation, and thus making the study transparent and reproducible. So let's see how we can start thinking about that. There are 4 main ideas, 4 main tips that we will cover in terms of quantitative data. Plan for reproducibility before you start the project. Keep track of things, that's really important. Share and license your research, and report your research transparently. Now we're going to go into more detail about each of these tips. So plan for reproducibility before you start designing the study. So create a study plan or protocol. Start with that. Begin documentation at study inception by writing a study plan or protocol that includes your proposed study design and methods. For specific guidelines on that, you can look at the Open Science Training Handbook. The link is in the bottom of the slide, and I can share my slides later with you so that you can follow links and find specific guidelines as well as tools suggested by the handbook for each of those steps. So it's important to track changes to your study plan or protocol using version control. And of course, before you start the study, it's important to calculate the power or sample size needed and report this calculation in your protocol, as we know underpowered studies are prone to reproducibility. Second is to choose reproducible tools and materials. Whenever possible, choose software and hardware tools where you retain ownership of your research and you can migrate your research out of the platform for use. And again, if you follow the link in the bottom of the page, you can see tools that can help you with that work. And the third point here is to set up a reproducible project. It's important to centralize and organize your project management using an online platform, a central repository, or folder for all research files. GitHub is a tool commonly used as a place to store project files together or manage everything using an electronic lab notebook. So within your centralized project, follow best practices by separating your data from your code into different folders. And it's important to make your raw data read only and keep them separate from processed data. So the second idea is to keep track of things, every step of the process. So registration, pre-register important study design and analysis information to increase transparency and counter publication bias of negative results. There are many free tools available to help you make your first registration. These tools include AS Predicted, Open Science Framework, which is used quite a bit in psychology, registered reports. Even for if you're running clinical trials, there is a specific repository for that called clinicaltrials.gov. Again, for those tools and those websites, you can follow the link to find out more. It's also very important to use version control. Track changes to your files, especially your analysis code, using version control all the time. Documentation is key. Document everything done by hand in a README file. Create a data dictionary. You might have heard of it as codebook as well, to describe important information about your data. And of course, it's quite good practice to literate the programming. Consider using different tools such as R Markdown. If you're using R to run your analysis in your code, R Markdown is very, very helpful and easy to use. But other approaches are out there too. So use those tools to literate programming, to integrate your code with your narrative and documentation. And that helps you again when you write up the paper or when you share your analysis to other collaborators. It's easy for them to follow your code and your rationale behind your coding decisions. Share and license your research, and on that you have to share data, materials, and software, notebooks, and containers. So in terms of data, avoid supplementary files, decide on an acceptable permissive license, and share your data using a repository. Again, Open Science Framework is one that is widely used in my discipline. In terms of material, you share your materials so they can be reused by other researchers, deposit them in repositories to make them easily accessible to other researchers. And in terms of software, license your codes to inform other researchers about how it may be used or reused. And finally, it's very important to report your research transparently. Report and publish your methods and interventions explicitly and transparently. And it's important to post your results to your public registration platform within a year of finishing your study. It's quite common, especially master and PhD students. They work on their projects for years, then they leave the PhD, they finish, they might go into industry, start working there, forget all about the research project, and potentially never publish that work. It's important to have a place, a repository where you can actually publish your thesis and the data so that other people are aware of your research and I can continue that. So this is just a resource that is really useful. This is the open online version of the book called The Practice of Reproducible Research. So this book contains a collection of 31 case studies of reproducible research workflows written by academic researchers in the data-intensive sciences. So each case describes how the author combines specific tools, ideas, and practices in order to complete a real-world research project. So there's lots of practical knowledge in the book, in those case studies, and can help anyone to start thinking about incorporating reproducible practices in their workflow. So emphasis is placed on the practical aspects here of how each author organizes their research to make it as reproducible as possible. One of the chapters on the book also is called the basic reproducible workflow template, which actually offers a template. I think it's on regression analysis, but it's something that beginners can start with to think about how they can structure their own reproducible workflow. So let's move now to qualitative research and the limits of reproducibility. So Sebastian Karcher, who's the Associate Director of the Qualitative Data Repository, has raised the issue of terminology problems surrounding reproducibility discussions. So more specifically, there are three terms that are quite often used interchangeably. We've talked about reproducibility, which is using the same data and methods to produce same results. We also have replicability, which is using same methods, but different data and different sample and arriving at same results. And finally, we have transparency, which is the process of providing all necessary information to evaluate a study. So Karcher argues that reproducibility and replicability are based on a positivist view of science, testing a hypothesis in a lab, and fits quite well quantitative data, whereas qualitative research may follow interpretative research methods where the study conditions are quite impossible to recreate. So therefore, Karcher suggested that the rigor and quality of qualitative research should not be judged based on reproducibility or replicability, but by the transparency of the research process. So in qualitative research, we will more likely talk about transparency rather than reproducibility. And there are three types of transparency most relevant in qualitative research. It's the production transparency, which is information on how data are collected, generated, such as research questions, sampling, subjects, recruitment. This information should be recorded following best practices in the research field. We have analytic transparency, which is the documentation process of qualitative data preparation and analysis leading to the conclusions of the study. And finally, we have data access, which includes considerations with regards to data sharing and access conditions. So, however, the idea of using transparency as a way to evaluate the rigor of qualitative research is only the beginning of a debate and not a solution for all aspects of qualitative research evaluation. So following the path of transparency is not always straightforward. So data preparation, data anonymization, and providing access to data can be costly, both in terms of time and resources. And there also might be ethical and legal challenges with respect to privacy regulations and additional copyright implications of textual data. So here are some of the three practical issues that are related to qualitative data. These are the identification of personal data, access controls for the data, and informed consent forms. And these are all important when you're dealing with qualitative data and qualitative research. So the identification of qualitative data is highly dependent on research questions and context. So local circumstances also need to be understood in order to know what gives away the data subject's identity. And we also need to think, of course, about minimizing the loss of richness of the information provided when we de-identify the data. And it's pretty clear that there is no model answer. Each project has to develop their own practices. For instance, in education and cognitive psychology that I work in, it's very common to collect data on participants' birth date, and usually we work with children. We collect the date of birth because it's important for us to know at which developmental stage the participant is when they are participating in our research. But of course, when we would think about identifying the data and sharing those data sets with the public, if we wouldn't publish the date of birth of the children, because that would make them very identifiable and quite easily. So we would publish their age in months or years, but we would have to really protect some characteristics of our data and protect the children. So it's important for each project to clearly and very think from the outset about which areas, which data they can publish and which not, and how they can protect sensitive data and that make participants identifiable. It's also very important to be aware of access controls for sharing qualitative data, which often contains personal information. And therefore, despite the fact that research data contains information which could potentially disclose individuals, their identity can still be protected through access controls. Some examples of access control to research data which can be offered by research repositories are conditional online access, so data can be accessed online but only by registered users. Depositor approval access, the data can be accessed online by registered users who are in addition approved by the depositor. Offline access, data sets can be accessed by registered users at a secure location on a computer with no network access. And embargo data, data sets can be only accessed after a certain date that the conditions need to be specified and justified for that. And finally, it's very important to understand the guidelines on writing clear and informative text in consent forms. Getting consent right is important to make sure that data collection is efficient, compliant to regulations, and that there are considerations of a potential reuse of research data. It is also important for researchers to negotiate with ethics committee on the text of the informed consent and sometimes receive advice from them to avoid risk-averse decisions by the committees. So the take-home message is that there is no one-size-fits-all solution when it comes to responsible working with qualitative research data, and that contextual information is often key to inform those decisions. So that said, in all cases, transparent documentation of all the data sets and processes are very important, as well as getting appropriate consent and ensuring adequate access controls to data. So I have a few more slides on examples of transparency on qualitative research. This one here comes from the economic researchers Aguinis and Solarino, who performed the literature search to develop transparency criteria for qualitative research. And they developed 12 transparency criteria covering research design, measurement, data analysis, and data disclosure. So anyone who's interested in starting thinking about transparency in their qualitative research, they can start with those criteria and reflect and see how they can improve their work. Another paper here is a framework for thematic analysis, which is a type of qualitative analysis, and how this can be made more rigorous and replicable. Can say more transparent. And finally, this is a template for pre-registering qualitative research, which is found on OSF. So that's all from me. Thank you for listening, and I'm happy to take any questions and of course share my slides with you so that you can follow the different links and get more resources.